We've come a long way in how we treat type 1 diabetes. We've also come a long way in how we understand type 1 diabetes. But what happens when the advancements in our treatment don't line up with our understanding? Welcome to episode 51 of Diabetics for Diabetics Radio. I'm your host, Bowie Madison. Today's episode is titled Exploring the Fine Print of the Newest Type 1 Diabetes Breakthroughs. For decades, treatment has centered around one assumption, that the immune system is broken, that it mistakenly targets the pancreas, that insulin therapy is the only viable way to step in for that damage. But what if that's only just scratching the surface? In this episode, we'll continue unpacking a new lens on type 1 diabetes, one that views the immune response as reactive, not rogue, one that asks, what conditions precede the attack on beta cells, and what terrain makes these cells vulnerable in the first place? We'll explore what's new in our understanding of diabetes development and how it challenges the foundational assumptions baked into every treatment protocol today. This includes the so-called cures, biotech promises like Biotechnology and Vertex that offer functional beta cell replacement. But at what cost? And what do these therapies actually solve? And what are we ignoring in the process? Because when you place new cells into a body still full of unmet needs, like nutrient imbalances, oxidative chaos, poor immune discernment, you don't always get healing. Sometimes you get compensation, sometimes you get a new problem entirely. Like immunosuppressive regimens that silence the very system meant to keep cancer in check. Like nutrient-deprived cells forced to adapt in unnatural ways. Like a metabolic environment still incapable of supporting life, not because it lacks insulin, but because it lacks balance. Today's episode is a call to go deeper, to question not just what we're treating, but how and why. Because true healing doesn't come from inserting new parts. It comes from restoring harmony in the whole. So here's what we cover in today's episode. Why do we treat diabetes the way that we do? What are the assumptions that we're making in how the disease develops, the systems involved, and the steps we think need to be taken to address those steps? And with what we're learning about disease development, are the cures matching what we're learning and all the systems involved? And so what's new with type 1 diabetes development? Are we paying attention to the multiple systems involved, whether that be the pancreas, the liver, the gut, the nervous system? We are kind of coming to realize that the immune system may be a secondary culprit in beta cell die-off. It may be what's holding the murder weapon, but it wasn't really what pulled the trigger. And then when it comes to looking at these new cures from Biotechnology, Vertex, and there's a litany of others in the pipeline now, what are they doing for our current understanding of diabetes? What are they targeting? What are they marketing themselves as in the context of our greater understanding? And if they're not doing the whole job, are there weaknesses? As consumers of these health products, are these things that we should know about? You know, how serious? What do we need? What more do we need to know about these to make sure that we can be protected as we consume what they're offering? And as we'll find out, there are quite a few. And what are some of the unexpected byproducts of these unmet needs, the things that these presented cures aren't covering in terms of disease pathology? We'll talk more about the dangerous combination of malnourished cells and immunosuppression. The next few slides, we're going to be branching off from episode 50. And if you haven't watched it, I highly encourage you that you do. We do a more all-encompassing look at the studies themselves from Biotechnology and from Vertex, and transposing that over a little bit more of a deeper breakdown of where we are right now as those with diabetes. We'll touch on it here, but this is all really a continuation of more of the deeper stuff that we learned in episode 50. First and foremost, I think this is probably the most important thing you could learn from episode 50 is that diabetes is a symptom, an end result of deeper foundational imbalance. And if you look at this graphic over here, diabetes is what we see at the surface, right? No beta cells, no insulin. We have to pay attention to what we eat, and that's it, right? And so if that's all we can see above the surface, that's kind of, that's what we think of when we think of a cure, right? If diabetes to us is beta cells and no insulin, a cure for diabetes would be beta cells with insulin. But when you understand that there is a underwater portion of that glacier, things like liver toxicity, gut health, nervous system, immune system, food, nourishment, a real cure that's going to address everything, both what we see and what we don't, you really have to pay attention to the roots of those kind of end stage symptoms, which diabetes is. And that it's just one part of the equation. And taking more from episode 50, these are some of the technologies being employed by these diabetes breakthroughs. This one specifically is from Sana Biotechnology, which is the immuno-evasive beta cell that you do not need immunosuppressants for. They do this by removing HLA expression. So this is a way for beta cells to be camouflaged from the adaptive immune response, in which antigens are presented to T cells to be judged as either friend or foe. And this takes that antigen away so that the T cells have nothing to read the cell by. And so it kind of bypasses that adaptive immunity. And then you have the CD47 signal. This is a cell marker that cells can release that let the innate immune system, the natural killer cells and macrophages, to let them know, hey, listen, we're not something you need to worry about. We're safe here. You can leave us alone. We're safe to exist. So you have these two components, two beta cells, that's preventing immune intervention. But those two technologies, the decreased HLA and the increased CD47, is an incredibly important part of how cancer cells thrive in their hosts, because it's a really genius way of evading both parts of the immune system, the adaptive and the innate, and how this is worthy of attention. Again, we're not entirely sure exactly how that plays into a healthy system, right? Just because cancer uses it doesn't mean it's inherently bad, but we need to pay attention to the details on this one, just because of what we know about cancer and how powerful it is in the body. And we also learned that the context in which these two technologies are being used by Sana are neglecting the foundational needs of the cells themselves, like nutrients, enzymes, cell signaling. They're not addressing these needs in the host, right? They're putting these cells in with this immuno-evasive technology, so they can't be touched by the immune system. But you have to wonder, do they have what they need to function for an extended period of time, right? Because, it's not just like beta cells don't have metabolic waste and they don't have nutritional needs, things like magnesium, calcium signaling, cell membranes, amino acids. These are things that every cell needs to function. And if we're not supplying them, what happens to them? And so is this cause for concern? Like, what happens in an untouchable cell that isn't being nourished properly? And even if they are being nourished properly, is there something that we need to know as consumers of this cure to help them stay nourished for longer periods of time. And then, anytime you talk about some kind of like cancer-born technology, you have to wonder, like, okay, well, if these beta cells are always on and can't be touched, is there any way to turn them off? And it turns out there's an anti-CD47 technology, something that kind of removes the invisibility cloak and allows the immune system to engage on the cells. Albeit Sana Biotechnology wasn't really forthcoming with this facet of the process, but there, if anything in those cells that can't be touched by the immune system goes wrong, let's say one of the cells, and they're injecting like 25 million, 60 million cells. What if a handful of them aren't doing what they're supposed to, right? It may not seem like a big deal in the context of, oh, well, if 99% of them are okay and 1% of them isn't, that's fine. But if that 1% can't be cleaned up, does it eventually become a problem? And the anti-CD47 technology is not cell specific. So if one goes wrong, you kind of have to take care of all of them in order to prevent one from ruining everything. And that's not really great. And it opens up the door to the fact that if every beta cell isn't just right, and you have to kill everyone off once one goes wrong, do you have to keep going back for new injections? And so it adds in some ambiguity as to the longevity and the permanence of these transplants. And we touched on it before, but there are a lot of questions coming from these solutions being presented. One of which we touched on before: how are these beta cells self-regulating in terms of their proliferation, their cell life cycles, and metabolic demands, right? If we've all of a sudden got all these brand new beta cells kicking on full cylinders making insulin, do we have the nutrients to make sure that this cell can sustain itself? If it eventually does need immune activation, and we'll learn, in a cell cycle, the immune system is there to kind of clean up the mess, even if it isn't like dangerous. It can be like a maintenance kind of thing to ensure that everything stays on track. If it can't do that, it can't clean up the mess. And if the mess gets to be too big, we're getting into things like tumor permissive environments where it just can't be touched. And this is kind of where that cancer connection comes in. And then the timeline for cell viability and life cycle length. We are all very excited about these technologies because they're hitting the 6-month, the 1-year mark at being viable blood sugar regulating, insulin producing cells. But what about in the future, right? Is this something that we're going to have to come back to and rehash? What are the side effects, the systemic side effects of these technologies? So I wanted to touch just briefly on what the innate and adaptive immunity does, right? They're typically not necessary in a healthy, self-sufficient cell, a well-supplied cell. It's really only when things go awry. And you'll see here in that first bullet, HLA molecules present on almost all cells, right? These are kind of like the little markers that T cells read and are like, are you friend or are you foe? If you're foe, you get knocked out. If you're friend, you're safe. These are situational sentinels to the immune system on whether a cell needs to be addressed, right? Situational is the specific word here because a cell that's healthy 1 minute might not be healthy the next based on certain factors like redox imbalance. Is this cell accumulating ROS faster than it can clean them up? Is this one cell threatening the livelihood of the group of cells? Are there misfolded proteins? Is there proper nutrient flow and electrolyte flow, fluid balance? Are the cell membranes intact? Is anything spilling out into the open and eliciting an even greater immune response, right? We need that's where the immune system really comes in handy, even if the cells as a whole are healthy. And touching on that redox balance, oxidative stress within the cell increases the expression of HLA actively. So if a cell is starting to have trouble inside, it starts to kind of send markers like, I think I'm going to be sick. Immune system, can you please like take care of me, please? And that's like a good function in a healthy system, right? Cells able to kind of self-regulate themselves to be taken out for the good of the whole. But any small scale hygiene, right? If we've got a perfectly healthy cell that has everything it needs, it doesn't really need T cells, right? Because everything's being processed internally. All of the ROS is being handled. All of the nutrients are moving in and out of the cell membrane. We're good. And then when we go to the kill switch technology, the anti-CD47 technology, what's the success rate, right? Like with this kill switch technology, is it every cell every time? Is this anti-CD47? Because CD47 is used by other things in the body as well. Is it messing with anything with that? Is our immune system kind of confused? Coming from people with diabetes, our immune system is already in a state of fatigue as it is. So do we know anything more about this technology? The timeline of it, right? Longevity and the permanence of those transplants. How long does this last? How long do we have before we might need another transplant? I don't know. And this kill switch technology, this anti-CD47, so removing the invisibility cloak or removing the safe signal so that the immune system can recognize it as not safe, it's not new, right? This is actually one of the key movements in the anti-cancer research realm. They're finding ways to eliminate CD47 signaling in cancer cells, but also in specific T cells to ensure that T cells that are specific to certain cancers can remain without being acted upon or broken down. So it's a way of kind of like using fire to fight fire. You're giving T cells the same technology that cancer cells have so that T cells can target cancer. So it's not entirely new. I think it's kind of ironic that it's being used in cancer and no one's making that connection between cancer and diabetes. But here's a quick little illustration of how that works, right? So to my left, we have this immunoprotected graft of beta cells. All these marked with asterisks. They're healthy cells. They're making insulin. Everything's good. The brown and the red cells, though, they're starting to like hiccup. And again, this could be a random gene mutation. This could be something related to cell resources where it just isn't doing what it needs to. It can't get what it needs to in order to function at a high level. These red arrows are the immune system, and they can't get in to like clean it up. So what happens to a cell that can't do what it needs to, right? It becomes a drain on the system. And if you can't clean the drain, eventually it gets clogged, right? So when you do the anti-CD47, you remove that cloak, right? The gray square is gone, and now everything is kind of exposed to this great big sweeping immune cleaning. And so that kind of clears out not just the brown and the red, but also the green healthy cells. And there's more to this, right? The HIP here, the HIP engineered cells, that refers to hypoimmune protection technology. And that's what it's called, the HLA and the CD47 combination. You can't just clean out the old and the damaged cells. It cleans out everything. And when you clean out everything, but you still need the beta cells that were there, you have to reimplant them, right? So this is a repeating delivery procedure. And there's no selective way to uncloak just the dysfunctional subsets. It's all or nothing. And so now let's move on to the shifting understanding of diabetes pathology and diabetes therapy. How do they match up? How do they line up? And what are we missing uh, between the cracks? So this is how we saw diabetes before. These are the two factors we were concerned with. We had healthy beta cells. Something happened. There was a triggering event. Our immune system went rogue. And now we've got auto antibodies killing off healthy beta cells. It could have been our genes that turned on or off outside of our ability to influence them. And now we've got dead beta cells, right? So our timeline, pretty straightforward, right? No diabetes, healthy beta cells. You add in this destructive immune response, and then boom, all the beta cells are gone, and we've got diabetes, right? Pretty straightforward, pretty simple to follow. But it's changing. Now we see diabetes as multifactorial. We have the beta cells themselves struggling. This is before the immune system even becomes involved. This is just not enough raw materials in the cell, dysfunction within the cell, from a cell infrastructure integrity, from a cell signaling. It's broken, it's breaking from the inside, and it's calling for help from the immune system outside. There's also outside inflammation, outside systems, right? Liver toxicity, our gut, leaky guts. Everything has become so inflamed that everything that beta cells were relying on for stability before aren't able to do their job. The beta cells are kind of have this result in instability themselves. And the immune system is just taxed. It can't do everything all at once all the time. So it doesn't have enough resources to properly clear the ROS toxicity and restore healthy tissue. And so now this is kind of our new timeline, the new progression, right? You have this poor cellular environment through a number of factors. And again, the list I just gave you isn't comprehensive. There are more factors and layers within each of these bullets, but something's going wrong in the cell. Something is going wrong systemically, so that beta cells are eventually struggling, asking for help from the immune system. The immune system comes in and is like, man, these beta cells aren't going to make it. I have to clear them out for new beta cells to come in. And diabetes results when these killed off beta cells don't have the resources to sustain a healthy volume. So you clean out the ones that can't keep up, but you don't even have enough resources to make new ones. And so you're kind of left without any left or enough left. And you have to take into account, given this new understanding of diabetes, what that existing state is, the pre-existing state that leads to diabetes, right? You have high oxidative stress. This could be through free iron accumulation. I argue that that is the case. We have a lot of free iron in our body that is leading to higher and higher oxidative stress levels with a decreased ability to manage them. You have acidic tissue environments. Beyond just diabetic ketoacidosis, we have increased lactic acid, glycolytic environments that can't really use glucose effectively, and it's leading to a lot of toxic metabolic byproducts. You've got calcification and the redox imbalance that results from an acidic environment, right? The blood becomes too acidic, bones release calcium to help rebalance and bring that pH back up. There's a lot of free calcium floating around, and the bones are softening, the tissues are hardening. And that's bad news when it comes to hormone signaling, like with insulin. And then immune system dysregulation. It's not rogue, it's confused, it's undernourished, and it's overwhelmed. It can't possibly do its job because everything's on fire. So this is where the new technology enters, right? And again, if you want to know the greater, finer details of this, head back to episode 50. But we've got Sana Biotechnology and Vertex Pharmaceuticals. Insulin producing beta cells with a regulated blood sugar within a 6-month to 1 year period post operation. Okay. By one line alone, that's awesome. We love healthy beta cells. We love insulin production. They are protected, right? Either the cells themselves prevent immune activation, that's the Sana biotechnology, or you have to use immunosuppressants alongside the transplants to prevent immune activation so that the beta cells can remain untouched. Neither of them are supplying any additional components to address the nutritional needs and the environmental needs of those beta cells, what I would argue caused the beta cells to flounder in the first place before the immune system came in. So the cures are basically saying, here are these beta cells. They make insulin, they regulate their blood sugars, and the immune system doesn't disrupt anything, right? And on the old timeline, this is outstanding. This is everything we needed because we had healthy beta cells and the immune system was the problem. So now that we took the immune system out of play, healthy beta cells now have the chance to repopulate and sustain. Everything's kind of, back to normal again. But what happens with this new understanding, right? Poor cellular environment, systemic struggle, liver, gut, nervous system, pancreas. The beta cells are like barely spitting out any insulin because they're struggling, and because they're struggling, the immune response comes in to clean it up. And so before you had dead beta cells without any resources to sustain healthy volume. Now with these new implants and no immune activation, you've got new beta cells that can't be attacked by the immune system, but they are still without the resources to sustain a healthy volume. There's holes there. So does this help with nutrient availability, organ system health, pathogen clearance, ROS and oxidative stress clearance? No, right? None of these cures are being presented with any sort of lifestyle modifications or even letting people know, like, hey, listen, these are ways to help sustain beta cells. It's just, here's the beta cells, should be fine in the system you've got, even though it didn't work so hot for the last beta cells you had. The immune system isn't the only problem, right? But if we're operating on a simpler understanding, an older, outdated understanding, then it's a fit. And they're just kind of going by with the assumption that that's enough and that's it, and hoping that nobody asks any further questions like what we're talking about here. And as a consumer of this technology, we have to balance like what we're willing to accept. Because yes, in the short term, I like beta cells that produce insulin. I like having normal blood sugars. And I like not having to worry about my immune system interfering. But am I protected from the pre-existing deficits that made my beta cells not viable in the first place? No, that's short term. That's kind of like a neutral, like, I hope everything turns out well. I love how it's been going this first year. I'm not sure what happens in the long term, right? And these are all question marks. These are not accusations. These are all questions from me. Again, this technology hasn't been around long enough to have any of these questions answered. But how long does that insulin production sustain? How long do those implanted cells survive without addressing the baseline needs from a nutrition standpoint? If we don't know that, we don't really know how long we have normal blood sugars. Again, you're protected from the immune interference, but as we come to find out, that may not be a good thing either. And then this systemic integrity. Our liver, has that improved at all? Have we done anything to improve it? What about our gut? What about our nervous system, right? So even if the beta cells themselves are protected and producing insulin, if you've got a bad liver, a bad gut, and a bad nervous system, you're working on borrowed time, because that's just going to present someplace else. You need the liver, you need the gut, you need the nervous system. And so does this make you vulnerable to new disease states? Because those side effects, oh boy. Cells numb to the immune intervention, right? Because they're camouflaged and marked as safe. They're using the same stealth tactics as cancer cells in a nutrient deficient terrain that has a history of not sustaining cell life before. It's not good, right? Because you can't not address the cellular environment. That's what the cell is pulling its lifeblood from. So there's no internal recycling system within these cells. How do they sustain themselves? If any one of them needs to be replaced or sustained or corrected, are they able to upregulate HLA? Do they just have no HLA expression? There's no internal safety mechanisms. Like, what if the cleaning needs of the cell supersedes the cell's ability to self-clean? Right? That doesn't mean the immune system's going to come in and just ruin everything. It's there to help. It's there to help clean. How do we coexist? How do these two things coexist, right? Like a cell that might need some help, but also doesn't get wiped out every time it needs a little bit of help. And if you have a cell that does need help and doesn't get it, what does that do? It increases its exposure to oxidative damage, which opens it up to DNA disrepair, mutations, and just the fact that the cell can't do what it needs to anymore. And Vertex, most of the hypoimmune technology that I was mentioning with the HLA and the CD47, that's specific to Sana Biotechnology and their HIP technology. But Vertex achieves the same environmental conditions just via a different method. You're still getting those beta cells. They lack the HLA downregulation and the CD47 upregulation, but you're taking immunosuppressants alongside them. So you're still silencing the immune system. And this has systemic consequences, because now you're not just isolating the immuno-evasion to beta cells. It's like your whole body's immune system is being targeted. And so, holy moly, what are we opening ourselves up to? And it turns out quite a lot. There's a lot of risks of prolonged immunosuppressant use, right? Increased infection risk. You've got things like Epstein-Barr virus, cytomegalovirus, HPV, human papillomavirus, all viruses that increase your risk for, number 2, cancer, right? Latent infections reactivating. Lyme's disease. I had a tick bite before that was kind of my triggering event, right? So are you just opening up a whole new can of worms in a body that isn't able to protect itself anymore? And with the cancer, tumor permissive conditions, again, you're decreasing HLA expression. Things that should be getting caught in the filter are not getting caught in the filter and just continually recirculating. It's not good. Not good. Neurotoxicity and cognitive side effects, right? Headaches, seizure, confusion, mood disturbances. Again, things that need to get filtered aren't. Metabolic dysregulation, hypertension, hyperlipidemia, insulin resistance, increased fat retention. You would think we're talking about something that increases your risk for diabetes, not something being used to help prevent diabetes, right? Like, hey, here's some healthy beta cells, but you got to take it with something that's going to increase your insulin resistance. That's insane. That's insane. And the list is longer than 6. There were more, but it's just like, you get the point. Bone health, muscle wasting, right? Decreased lean muscle mass. Kidney and liver toxicity. Remember, we were talking about how important the cellular environment is for beta cell integrity? Increased or prolonged immunosuppressant use is making all of those pre-existing conditions in the diabetic state worse. It's like one step forward, 6 steps back. No sense. So you have to ask yourself, is the juice worth the squeeze? The immunosuppressants can buy time, right? And a lot of times in transplants, it's like giving the transplanted cells the ability to establish themselves. And I don't know if there's some kind of like weaning off process where some of the cells can eventually achieve tolerance, without permanent immunosuppressant use. Because if you use immunosuppressants too long, it comes at the cost of removing the layers of self-regulation, immune defense, and metabolic equilibrium that you need to be healthy in the first place. So these are not non-trivial trade-offs. They have to be balanced with the use, right? You don't want to take a cure in one hand in exchange, like, and have a new disease in the other hand. It doesn't make any sense. And I bring up cancer because it's such a common thread. It was on the forefront of my mind because I saw the ties between low HLA expression and high CD47. Unstable cellular infrastructure and signaling. There's a whole checklist of things that cancer thrives in, right? And then I wondered, okay, well, of all these things that are kind of like tumor permissive and pro-cancer, how many of those things exist in the pre-diabetic or the diabetic state? Does any of this mess with the kill switch technology, the anti-CD47? And it seems strange to me at a baseline level to kind of borrow this very influential cancer characteristic in another health facet, like we know what we're doing with it. It's like, oh, if you know what you're doing with the things that make cancer work, what's going on in the cancer research world? It's really, really strange. So let's think about the cellular terrain in a diabetic state, right? A diabetic body, I would argue, and again, you can look back at my portfolio of podcast episodes and research materials, we're way too much iron, way too much free iron. Enriched foods, our iron retention reflex is always engaged because we are living in this chronic state of inflammation. Nervous system hyperstimulation, we're burning through magnesium. We have trouble with fluid retention, poor digestion as a result. It's really hard for parasympathetic tone. Compensated breathing, right? This can lead to CO2 buildup. This increases our body's acidity. This acidity messes with our calcium signaling, right? Leads to bone resorption, tissue hardening. With our food the way it is and the degree of inflammation we're experiencing on a daily basis, we just don't have the antioxidant capacity to handle for the degree of oxidative stress that we're going through. Part of it has to do with poor liver function and gut instability, right? If our main filters, the main things we use to filter our blood, filter our food, and make sure that we can absorb things are broken, none of these other things stand a chance. And I really don't think that any one takes the cake over the other. These all kind of like synergize together as a system drain, and it's really, really, it's just a matter of tracing which one is, picking them apart piece by piece, to help the system get back to where it's supposed to be. But this is kind of what we're thinking of for those with diabetes. I think if you have diabetes, type 1, type 2, anything in between, we're experiencing some degree of each of these. And so if that's the case, and we're thinking about cancer and diabetes, we got to kind of look back at what were the things that were good for cancer before, right? This is the slide we looked at before. High oxidative stress, free iron accumulation, acidic tissue environment, calcification and redox imbalance, and an overwhelmed immune system. Is that pro-diabetes or is it pro-cancer? It's both. If I had told you that, if you like hold your hand over the title of this slide and you ask someone who knew about cancer and knew about diabetes and had them have to choose which condition the slide was about, they couldn't tell you. It's both. It's both. And I'm not going to go through each of these slides like point for point, but this is serious because low pH, right? You've got high acidity, DKA in those with diabetes. The inflammatory state itself is acidic. And so this leads to insulin resistance. This leads to mitochondrial dysfunction and cell membrane integrity issues. These are all not good things, right? So everything here in relation to acidity is something that cancer thrives in, right? And I threw in paper titles to let you know that I'm not just making this up. I have links for all of these papers in the description, but do your own research. Look into this yourself. There is a lot of material going into what we're talking about here. So we'll talk about calcium balance, right? Calcium is a major part of how insulin is released. And when calcium gets dysregulated, insulin doesn't get released efficiently. And so you just have this huge cascade of the body's attempt to try and balance acidity, balance calcium in the body. And in the diabetic state, whether it be through insulin resistance, low magnesium, altered food, it's all putting us in a position that is very tumor permissive. Again, here's one of the quote or the resources. Low magnesium. Again, this kind of goes hand in hand with that calcium dysregulation. So low magnesium, this could be hyperstimulation from the nervous system, leading to an increased magnesium burn rate. You need magnesium to help repair the enzymes needed to keep DNA stable. You need it for mitochondrial health. You need it for liver health. And if you don't have magnesium, you don't have any of those things. Not having any of those things makes you more susceptible to cancer. Link in description. Vitamin D, right? Everyone was talking about, there's a lot of papers talking about how unclear supplementing vitamin D is on cancer outcomes, but you have to look a little bit deeper, right? What about vitamin D receptor? What about our body's natural ability to create its own D and absorb its own D? So many things happening in a diabetic body are compromising our ability to create our own vitamin D, to absorb supplemented vitamin D, and this increases our risk for the big C. And then just a few more: redox imbalance and chronic reactive oxygen species, chronic sympathetic tone. All of these conditions coalesce to put you in a really tough spot when it comes to A, managing your diabetes, but also B, protecting yourself from the development of tumors. And I threw this one in here too because I just got done a few episodes ago talking about the growth hormones, IGF-1, human growth hormone, and testosterone in beta cell health and in the development of type 1 diabetes. And it's also got ties to uncontrolled cell growth, right? These growth hormones being applied in incorrect balance leads to improper cell proliferation, which is what tumors thrive on, right? This insulin is a growth hormone in and of itself. And for those developing diabetes where there's a lot of insulin, but you're also resistant to that insulin, the insulin does something. It's pro-growth somewhere. And so you're putting a lot of growth signals out into an environment where it's being directed towards growth that you don't want to grow. And here is this table. Feel free to pause the video here and kind of just take this in. This is kind of a summary of the dysfunctions that we see in those with diabetes and how they influence our risk for developing cancer. So pause just to take a deeper look. So now when you factor in that just having diabetes itself increases your risk for developing cancer, and you introduce something else that is specifically designed to create a condition where cancer can thrive, not a good spot to be, right? So you have all this new technology coming out in the name of preserving beta cells that ends up putting us in a tough spot when it comes to regulating cells with unknown growth abilities, right? These new beta cells, we don't know how they grow. We don't know how they proliferate or sustain themselves. But we do know that the immune system can't do anything to regulate it. So the tricks these cells are using don't just fuel the immune system to let these beta cells exist. They're teaching the immune system itself to ignore abnormal growth signals elsewhere. So you're suppressing senescent cells, right? These are old, aged cells. DNA mutations, damaged DNA, viruses having their way with these cell grafts. Again, the immune system has no way to intervene. In an environment pre-existing the new beta cells, where you've got a lot of oxidative stress, unstable methylation, free iron, an acidic environment, and diminished vitamin D signaling. What more could a tumor want? And I want to quickly interject here that I'm not bringing up this diabetes cancer connection to make you afraid. It is scary to bring up cancer, and all of these correlations, these similarities in conditions that bring about both diabetes and cancer got me thinking. And again, you're watching me realize all of this in real time. The similarities were just too strong to ignore. But it brings up a good point, because you may have already started to think to yourself, well, Bowie, if there is such a similarity in terrains between diabetes development and cancer development, why don't more people with diabetes develop cancer? Because again, I haven't looked up the stats on it, but it's not great enough to be at the forefront of our minds. Yes, people with diabetes are always told that they're to some degree immunocompromised, but it's not noteworthy. You know what I mean? No one in their onboarding when they're diagnosed with diabetes gets a cancer talk as well. And so when I thought back on that, okay, if the conditions that brought about diabetes are so strongly tumor permissive, as we've talked about just now, why aren't more people with diabetes getting cancer? And what if it has to do with how actually functional our immune system is? What if our immune system really actually is doing a good job? Again, this flies in the face of the autoimmune theory in which the immune system just doesn't work. But what if our immune system is doing a good job? It could either be doing a good job in regard to cancer and that it's very beta cell specific. That to me seems less likely, but what if it actually is just doing a good job against dysfunctional cells? This is another point in the direction of this is diabetes being a truly cellular based disease and not something based in a dysfunctional immune system. And I touched on this here. So the fact that Sana Biotechnology and Vertex Pharmaceuticals are not talking about how these implanted beta cells are sustaining themselves. Again, they're just kind of like, hey, they're not being attacked by the immune system. Yeah, but I mean, how do they grow? Do they self-regulate? What's their source of fuel? What's the integrity of their internal mechanisms like? Right? How does this work? How are they sustaining themselves? Immuno-evasive beta cells are not currently optimized for natural turnover, right? A cell ages, it dies, it's renewed. Just the cycle of death and birth. They are immune invisible, so natural clearance is hindered. Again, what is the degree of hindrance? Can they be cleaned at all? They may not have been designed with self-replication capacity. Again, we don't have a lot of data on that from these big tech companies. So how do these worn out cells, how do these undernourished cells, how do some of these defective cells get cleaned without a global kill switch that leads you needing an entirely new transplant? So you have to wonder aloud, what does this look like in a real world situation? Your body, in its state of diabetes, which is already fighting to regulate calcium, not just in beta cells, but bodily wide. Liver, gut, nervous system, immune system. Keep iron in check. Maintain the redox balance to allow for proper and healthy mitochondrial function. And then we take away its ability to flag a mutating cell as dangerous. It's really it's not a good situation to be in, because you're not in a safe space, and your ability to communicate that you're not in a safe space is taken away. It's breeding a lack of safety. Now, this is a lot, and it's something I'm pretty heated about right now, but at the end of it all, I am not against diabetes technology. I'm alive because of it, right? I've been a pump user, I've been a CGM user. They are all incredible things that I've used to manage diabetes. I'm not against them. But I have demands moving forward, because our awareness of the disease is ever increasing. And I need for the solutions being brought forward to match that understanding, and that's just not the case right now. So if these solutions, this beta cell replacement, is going to be part of the path forward, we need to start the conversation about the other components of beta cell health, beta cell stability, digestive health, nervous system health, cell signaling. Those need to be co-developed and added to the mix as ways to reestablish not just beta cells that produce insulin, but healthy beta cells that can sustainably create insulin over a long period of time. Right? So as we get closer to engineering a functional cure for type 1 diabetes, we cannot make the mistake of trading one chronic condition for another. Again, cancer comes up because of the hypoimmune technology that Sana Biotechnology is using. It is a powerful tool that, if it is not wielded responsibly, can be very dangerous. So we have to have the courage and the confidence as consumers to demand better. And if we come to the table desperate for answers, saying yes to anything that gives us what we want right now, we're forfeiting our power. We're forfeiting our autonomy and our health. And we're denying the progress that we've been making in understanding the condition. We're settling for something less than what we need. That is not acceptable. That does not play anymore, because we've realized we control our health outcomes through our education, our collaboration. And I don't want anyone to just melt at the thought of, having the tip of the iceberg being covered at the expense of exacerbating the underlying conditions and putting your future in doubt because you got what you wanted in the moment at the expense of your long-term health. And that's what I see is happening right now. So this is episode 51. I can't thank you enough for being here. We need to keep this conversation going. This is so much bigger than just diabetes. I want to know what you thought about it. I want to know what questions you have. If you see something that you want to talk about, let me know. I want to see what comments you've got for me. If you'd like to support the channel, subscribe, follow. I've got all of my links and resources available in the description. Your support is very, very much appreciated. And again, 51 episodes in, Diabetics for Diabetics Radio. I'm Bowie. Thank you so much for being here, and I'll see you next time.